Aside from inspecting the color of our samples by eye, we may actually use a spectrophotometer to get the real color of our sample. So we're going to test the colors of our samples with its absorbance with a color of light that has a 595 nanometer wavelength, or yellow. So the more or the higher the absorbance is, the bluer our sample is. So first, we are going to be inserting the cuvette with a blank sample into our spectrophotometer. And this is to calibrate our spectrophotometer at 595 nanometer absorbance for light. First, we need to wipe our cuvette first with Kim wipes. So this is sort of like a lens paper so that our cuvette will be smudge free. And then we also remove the parafilm before inserting the cuvette in the container here for the blank, labeled B in our spectrophotometer. So take note, when we insert, the side where the arrow on the cuvette is pointing down is exactly on this portion where there's a hole on the container. If you can see the hole there, that's where the light with a 595 nanometer wavelength passes through. So here's the process of calibrating. We are setting this 0 absorbance for our blank. And then we are going to do the same for our other samples. So here we have cuvette 1 for our standard number 1. We are going to insert this in the number 1 position here. And then cuvette number 2 with sample number 2. And then cuvette number 3 for sample number 3. And then cuvette 4 for sample 4. And lastly, cuvette 5 for this batch for sample 5. And then we get the absorbance. So here's for protein standard 1 or cuvette 1. So please record the absorbance value shown here. And then for protein standard 2, so here's its absorbance at 595 nanometers. And then for protein standard 3, and then for protein standard 4. So take note, you need to record these because you will be making a standard curve of the absorbance of these protein standards. And that you will be using that to estimate the protein concentration of your samples A and B, the milk samples. So here's the 5th sample. And then we remove the standard protein samples, but leave the blank behind so that we may insert samples 6, 7, and then our A and B milk samples. So here's sample A. And lastly, sample B at container number 4. So here's the absorbance value for protein standard 6. And then the absorbance value for protein standard 7. And the absorbance value for our milk sample A. And lastly, the protein, the standard or the absorbance of our protein sample B. And then we remove the cuvettes.